you believe that the enteric-coated capsules have the ability to bypass the copper complex issue completely, or is there still the possibility that it will complex small amounts in the patient's bloodstream?
I mean, it's not my belief. A delayed delivery is likely to reduce the degradation in the disulfiram, and that is known actually. And more delay is more less degradation because liver is the main organ, even though the upper part of the intestine also can degrade disulfiram. So degraded disulfiram binds with copper, not the intact disulfiram. So obviously, even when the degradation is inhibited or delayed, the ability for it to bind with copper and deliver that copper to the brain is kind of less when compared to normal. So enteric coating likely, as other people observed, is likely to delay the degradation of disulfiram.
Does the copper complexing occur in the stomach, or does it occur in the bloodstream?
Wherever there is a copper is there, form a very good complex. People eat with the food that is with a lot of copper, obviously, there they have a lot of degradation in the stomach, like lower pH, then it's likely they can form a complex there itself. But if it is degraded product is circulating in the blood, and then blood is, let us say, they have little higher amount of copper, then it is likely to form a copper complex there. And then, as I mentioned to you, disulfiram also have a very good tissue distribution. Wherever you have a lot of copper accumulation, it is likely to get into that place. In fact, there's one research paper. People used disulfiram for a tracer study of copper. So, as I mentioned about PET scanning is a method. And then what they do is they give a disulfiram injection to the patients, and they give a isotope of copper so that they can measure wherever the copper is going. And because copper in the blood complexes with degraded product of disulfiram, so after they give disulfiram, they showed that it can get to the brain. So you can use it, disulfiram, as a complexing agent of the isotope for PET scanning.
If the disulfiram gets degraded in the stomach, or if it gets degraded at any point, it at that point is vulnerable to complexing with copper. Is that what you're saying?
That's correct, yes.
So in its prodrug form, which is how it comes, it cannot directly complex. It's only once it's broken down.
That's correct.
And the acid is what causes it to break down?
Degradation, automatic in low pH, it is possible, little higher extent, but not complete degradation happens at low pH. It needs an enzymatic degradation pathway also.
So low pH and certain enzymes are what lead to the degradation of disulfiram. Does it degrade into any specific one molecule, or it goes into a whole host? And
once it is degraded, there are so many molecules, including carbon disulfide. In fact, that is the reason that the metallic taste and the smell people develop because of disulfiram.
Yeah, I've heard a lot of people saying they smell like garlic.
Yeah, that's because they generate a lot of carbon disulfide.
Would it be smart to at least not consume high copper foods with disulfiram? I mean, we talked about this last time. It's probably better to take it enteric capsules on an empty stomach. But if you were going to be eating, I would assume having copper in the food would probably not be a smart idea, having high amounts of copper in the food. Is that safe to say?
Yeah, I think so. But again, your dietitian or clinician need to advise you on that. I would avoid copper, yes.
Okay. Can you speak a little bit about why a liposomal formula might be better than a non-liposomal enteric-coated formula?
Found out that disulfiram absorption is better in the presence of lipid. When they eat fatty materials, the absorption they showed it's better. If you are delaying the delivery, you need to make when after the delay, like if it is released in the lower gut, it needs to absorb well, right? So perhaps that is how liposomal formulation is helping better absorption there.
So you did mention that disulfiram is a lipophilic or fat soluble or oil loving molecule, right?
That's correct. Liposomal delivery is kind of specialized technology. There needs to be a very interesting technology to protect these liposomes and make it intact till the place of its delivery, and this formulation need to deliver it to the walls of the intestine without getting dispersed in the food. It's kind of challenging task. So I'm not sure the people are already claiming that they have liposomal formulation have the technology to deliver either it is vitamin C, that is ascorbic acid, or disulfiram. I'm not sure about it. I have never seen any data on the efficacy of this liposome form.
Is there anything you can tell us about the new formulation that you are working on?
As I mentioned, it's a kind of involved technology of making a lipidated form, stabilizing them and then making it to formulate in a way that get delivered only in the lower gut, and make them to go to the closer to the intestine and specifically deliver the encapsulated form to the walls of the intestine. That is our technology.
You're working on a encapsulated form, not a tablet.
No, we are not working on the tablet. We are working on the encapsulated form.
Interesting. Do you know what kind of time frame would be expected for this drug to come to market?
It's currently developed by a company called Flight Path Biosciences, and maybe in a year, I don't know how much time they will take to commercialize it.
Do you know what are the regulatory things that need to be done? Do they have to do clinical trials, I'm assuming?
They are planning to start the clinical trial very quickly as possible, because there are a lot of enthusiasm about the slow delivery. So once they finish the clinical trial, maybe we can even look for accelerated approval for this, because the side effects of unformulated material is very bad. So I think we can convince the FDA.
What do you think the effect of disulfiram in an intravenous form would be? Would that be superior to an oral form?
The answer is I don't know, because I never put this animal to this tail vein injection or IV exposure. I think it's a worth studying it, and it's a very difficult experiment. It's not easy. We never tried so far. We always expose them with IP, and that paper we already published. But IP is not like IV. It's not like it is. IP is like you have a kind of intraperitoneal cavity. In that cavity, you are delivering disulfiram, and then the disulfiram is absorbed from the exterior part of the blood vessels to this blood. It's kind of different absorption mechanism. But we never gave directly the animal to the disulfiram and then try to see how the Borrelia is cleared, how it is getting into a different part. It's worth the experiment. We will do that perhaps very soon.
I assume that this formulation will help to reduce all of the major side effects like neuropathy, liver toxicity, psychosis.
That's correct. Yes.
So the liver toxicity is due to the copper complexing, right?
No, the toxicity happens because surge of degraded product in the brain and other important organs. That's the reason. It's not like a sustained release of the degraded product. It is sudden increase. So there is something called mass effect, mass action principle. That means if you have a very high amount suddenly increased. That's what happens in effervescent tablets. Like you take effervescent tablet, suddenly there is a surge of concentration. And also in this particular case, it's metabolites. Because the metabolism of the degraded product of disulfiram, it never gets saturated because many things metabolizes it into pieces. So you have a lot of degraded product production at the same time. So that makes the side effects very unbearable. The technology is try to have a sustained release so that we can control the metabolites formation by specifically a targeted slow delivery.
The psychosis effect, that's due to the inhibition of dopamine. I think it's dopamine hydroxylase, if I recall correctly. And from my understanding, that's related to the copper. So I just want to clarify, the neuropathy is caused by the copper. The psychosis is indirectly caused by the copper and how it affects dopamine hydroxylase. But the liver toxicity is not caused by only the copper. The copper doesn't help things, but it's primarily the surge of all these things that get produced when you do the first pass metabolism. The surge of all these metabolites and the strain that puts on the liver when you get hit with all of these milligrams of the drug at one time. That's what's causing the damage to the liver, right? Not the copper itself.
We don't know actually. In fact, the toxicity mechanism of disulfiram and its relation to copper, that itself is the subject of one paper.
Okay, so you're not 100% sure what is causing the liver toxicity?
No, so as I mentioned, toxicology of a drug is a very, very involved subject. Of course, one of the reasons we intensified this program in my lab is to study thoroughly, just liver toxicity and also the brain toxicity, neurotoxicity, and peripheral toxicity, whether it causes any toxicity in the heart. Everything we are simultaneously studying.
I saw a study that was done in, I think, 2001 that had showed, or it might have been a little later than that, but it showed mitochondrial impairment, potentially permanent damage to the mitochondria from disulfiram. Did you see that study by any chance?
Yes, I saw the study. In fact, we wanted to check the mitochondrial integrity in the animal treated with disulfiram. We will have some answer very soon. But I so far, whatever we saw in our preclinical model, it's not appearing like even disulfiram toxicity, anything permanent so far.
Interesting. And then you mentioned potential cardiotoxicity. Is that?
Yes. So we are very fortunate to have a human cardiac model actually, where we generate different kind of beating heart, a smaller micro heart, tissue organoid in the dish that's available with us. So we can check how the signaling changes by first exposing them with the boreal product, and then later trying to treat with disulfiram. We just proposed these experiments in one of the grant currently I'm writing.
This drug has been on the market for so many decades. How could there be all these issues with it that are not very well known? I mean, I know the neuropathy one.
The reason for that, this is not an issue for normal people. Only for patients who had an exposure of Borrelia, they seems to have problems. So the toxicity itself is very, very specialized mechanism that happens only in patients who had an exposure with Borrelia. In fact, that is one way to check if they are more sensitive to disulfiram. It's likely they have some kind of similar pathological mechanism that is operating in borreliosis. We can indirectly say like that.
That's, I guess, a way to check whether or not you might be infected, theoretically. S-
Yes, yes, yes.
So, this new formulation that you're working on, number one, it's going to be easier on the liver because it doesn't hit the liver all at the first pass
That's
metabolism.
correct. That's correct.
Number
Yes.
two, it's also not going to complex with copper as much because it's going to not get into the stomach acid. It's going to be released in the large intestine, which prevents the degradation, which causes all of these problems.
That's
The copper complex, the other complexes are all due to the degradation, and if you can release in the small intestine, do you completely stop the copper complexing issue completely if you get that far before getting hit or opening the capsule rather?
I mean, totally avoiding copper complexing is very ambitious. I don't think anybody can achieve it with existing disulfiram molecule as a drug. But we can reduce it. We can reduce it to the level the people, the side effects is kind of reduced much, much lower level. We can't tell that there will be a zero copper complexation by this method.
Okay. Well, it sounds like you're at least sure that it reduces it significantly.
That's correct, yes.
I wanted to mention there was a study that Dr. Lewis did that found disulfiram was not very effective. Did you hear about that study by any chance? His mouse model of disulfiram failed to eradicate Borrelia, apparently.
I don't know. I haven't seen any published paper like that. So we do have a lot of failed formulation. In fact, I have a formulation which increases the side effects so bad. This you can literally see the animals are they felt very bad actually because of the side effects. So that data we put along with the positive data just to show how important a delivery of disulfiram.
Out of
So,
curiosity, could you tell me what was the one that was very bad? Why was it very bad?
so what we did is we developed a formulation that is likely to be absorbed very effectively to the liver. Those animals, they had a higher level of toxicity, both liver and also brain toxicity because of direct delivery to the liver. What I wanted to show by this paper, in case if it is totally getting to the liver, 100%, let us say, close to 100% get into the liver, what will happen? That is the question we asked, and we showed the side effects is very high. That is why we think avoiding first pass metabolism is very, very important.
Okay. So just a high absorption very quickly would probably be.
In the upper part of the gut
Yeah.
is very bad, yes.
So the small intestine is a no-no. You don't want to be absorbing there, right?
Yeah, that especially closer to the liver, yes.
Is the copper diethyl dithiocarbamate complex not only responsible for the copper-mediated neuropathy, but is it this copper complex that's also partly or wholly responsible for the liver toxicity that's seen in some patients?
We just initiating a liver toxicity assay with various metabolites I procured. I just received all of them. Now they are doing it. We will definitely publish it very soon. We will not hold any data. There is one particular area where the liver first part metabolism absorption happens. So the formulation need to be protected from absorption from that area. So what we do is we specifically formulate and we size the formulation in a way where the absorption, the intestinal absorption near the liver is maximum. That's possible actually. We achieved it. One enzyme called HO1. This HO1 is kind of, it generates a lot of bilirubin. That's why they develop jaundice. So we need to inhibit that particular enzyme in the specifically liver. But this enzyme is also there in the brain. And if you inhibit that enzyme in the brain, it is very toxic. So we face this challenge with my collaborator. My collaborator asked me, I want you to formulate to these young kids. It needs to be given orally, but at the same time, it should not go to the upper, lower part of the intestine. It needs to get absorbed only to the place where liver absorption is maximum. That was the problem given to me. And again, we will measure the brain level of this particular drug. You should have the 0 level. It should not get into the lower intestine. We took the challenge and we developed something totally targeted to the liver specifically. And it worked very well. In fact, one company licensed that product to develop for a pediatric, to address the pediatric jaundice, to prevent it.
You're trying to target the drug to be absorbed in a specific part in the intestine that is not the small intestine. Is there any specific part of the large intestine that is trying to target, or the whole thing is fine?
Already, drug delivery people like me, they worked colon targeting materials. In fact, plenty of them are available in the literature. And this particular case, it's slightly different from just targeting to the colon. Like, for example, if you wanted to treat the colon, like colon cancer, or lesions in the colon, or ulcerative colitis, there are ways to deliver in the colon specifically. There, the colon tissue need to be exposed. Here, in our technology, it needs to be absorbed in the lower part of the intestine, but the absorption need to happen so that it gets into the blood. It should not just retain only in the colon. That's the challenge. I think we addressed that with our novel formulation.
Do you know if disulfiram negatively affects the gut flora or the migrating motor complex?
No, the bacterial flora reverses. It's in a healthy way. People who are in disulfiram.
So I assume a patient who has an allergic reaction to nickel can take disulfiram?
I don't know answer for this, but one thing I know, if people having allergy with rubber, so there are data to show they will develop allergic reaction. That's not big no to those patients.
If someone's allergic to sulfites, I assume that's not a problem for disulfiram.
I don't know.
Can someone who has nickel titanium alloy hardware in the body take disulfiram?
Any heavy metal in the body is likely to have a lot of adverse effects because of disulfiram. Lot of metals it binds, not intact disulfiram, degraded disulfiram. Titanium is a metal actually. We are talking about salts, soluble form.
Okay.
Metal is basically, they will put like polymeric coating and all. Generally, they don't degrade and leach out.
So it's really the degraded disulfiram that would chelate.
That's correct.
Do you know if it is contraindicated to use disulfiram if you have a high heavy metal load or of mercury or lead?
There are attempts to use disulfiram as to remove the heavy metals. I think that is quite possible. The problem with the disulfiram chelation therapy compared to other molecules is disulfiram can also get fixed in the protein. So it's not like the complexes and, comes out.
So you're saying the disulfiram can get stuck inside the body?
Yes, but there are like very big metal poisoning. You need a very effective chelating agent, and even the fixing of this metal may not be a problem because slowly even the fixed disulfiram, it can degrade, come out along with the metal. But the hike of metal concentration, if somebody needs to remove it all over the body, because disulfiram goes everywhere, I think in that sense, it's a good therapy.
Do you know if disulfiram's metabolites strongly chelate metals? Would disulfiram worsen mercury poisoning?
I don't know that.
Do you know why there are some patients that experience extreme fatigue on the higher doses of disulfiram?
Maybe they are more sensitive to acetaldehyde because acetaldehyde causes a lot of fatigue.
What are the primary symptoms of acetaldehyde buildup or toxicity?
It causes a lot of pain, number one. Number two, fatigue, as you mentioned, acetaldehyde exposure or accumulation in the body.
What kind of pain exactly would it cause?
I will forward the publication from my university. Somebody looked at it very carefully when they have accumulated acetaldehyde, how the pain changes. I will send it to them.
The increase in fatigue from Herxheimer versus the increase in fatigue from a copper toxicity, are those distinguishable?
Yeah, yes, I do think it's distinguishable. I think we can tell because the Lyme patients, they describe pain in a different way than pain due to drug toxicity. Clinician, at least, they can recognize what causes them pain. Toothache and headache, people can differentiate it. Different organ specific pain, diabetic pain, people can differentiate them compared to cancer related pain. So same thing, I think the drug related toxicity and also the pain due to infection, I think people can recognize it, differentiate it.
Is the pain associated with treatment caused by inflammation from the Borrelia being killed, or is the pain just an overexpression of cytokines? And what can be done about those side effects? Since
maybe both actually.
Okay. Is there anything that can be done to lower the cytokines?
Yeah, there are molecules available to reduce the cytokine storm. We saw a less cytokine production, inflammatory cytokine production when you are when the animals are exposed with disulfiram.
Affects the nuclear factor kappa B.
Oh, NF kappa B, yes. And if it is inhibited, you have less inflammation.
You believe that disulfiram may be inhibiting NF kappa B?
It is some more information is needed for us to tease out the mechanism, how exactly it acts as an anti-inflammatory molecule. And in my lab, people are studying that very carefully. We are likely to come up with a manuscript very soon.
Do you have any idea why the neuropathy symptoms seem to last so long in some patients even after they've stopped the disulfiram?
Regaining of neural function is always slow. Again, we need to check how long, at least in animals, how long they take to regain all the neuropathy related issue. That's very important. I also have experience. I was having prolapse in my spinal cord, and then, in fact, I lost ability to walk for some time. I was kind of bedridden when I was I'm not, late 20s. And even at one point, they told that you have to undergo a surgery to fix it. But some of my friends advised me not to go for surgery. They said if you wait for a period of time with mild exercise, you will regain ability to walk. I started doing some good exercise. That really accelerated my healing process because exercise increases the perfusion and exercise also increases neuroregeneration.
If you could explain how the nerves are being damaged by the DETC complex.
Other people already showed the possible mechanism. They think the copper complexation and also it can covalently inactivate certain proteins in the nerve terminal. That is how they think it is acting. It's basically by exposing the metabolites of disulfiram, you have a lot of fixed ligands in the brain, and then these ligands can accumulate copper in the nerve ending. That's what other people showed actually in the possible toxicity. And we are also trying to look at it in that angle.
Is there anything that we can take or any foods that we can eat that would help lower our copper levels?
I will find out.
Well, some people are taking zinc. That much I know in order to offset the binding of copper. Does disulfiram vulcanize the myelin sheath similar to how it vulcanizes latex?
People originally used disulfiram to vulcanize rubber. You know, the rubber, you have a lot of sulfhydral bond. Disulfiram can make the sulfhydride bond to, covalently link with each other. That is how disulfiram people used. And then some people think that when it is getting accumulated closer to the neuron, and the neuron can, the neuron terminal can get kind of cross-linked if you have plenty of sulfhydral protein. And the answer is we don't know. And we find disulfiram is having a different property than what is already known in the literature. So it's better we access some latest paper and then try to educate ourselves on this.
Do you know why there is less neuropathy as a side effect when alcoholics take the drug versus when Lyme patients take the drug?
Alcoholics are not having perpetual inflammation in their brain and their body. But people with chronic condition, we don't know why they have the condition, but they do have some inflammation going on. So they are more sensitive to disulfiram, obviously. So that I think that is the reason.
You have two causes of neuropathy. One is just treating Lyme disease in itself, and then another is the copper toxicity issue, which is...
That's correct, yes.
Is there any way to control the dopamine levels from disulfiram being increased excessively?
Yeah, there are papers about it. I can share the paper with them.
Some people are reporting experiencing joy, happiness, and calm on disulfiram. Do you think that this could be due to the dopamine increase?
That's correct, yes.
Is the decreased libido also caused by this dopamine increase?
We are suspecting one particular pathway is kind of interfering. Maybe that is the reason. But, libido is also related to the stress.
Is the fatigue possibly due to damage to the mitochondria, or is it copper toxicity?
I don't know the answer, but it's worth answering.
Potential cardiotoxicity, is that?
Yes. So we are very fortunate to have a human cardiac model, actually, where we generate different kind of eating heart, a smaller micro heart, tissue organoid in the dish. So we can check how the signaling changes by first exposing them with the boreal product, and then later, trying to treat with disulfiram. The heart function of the people who are on disulfiram for several years, it's not, reducing. If that is correct, like if it in vivo, it can cause a lot of apoptosis, then people will be losing their heart cells, and then they'll be losing heart function, and so that never happens. So let me do more experiments. In fact, my position in cardiovascular institute, so it's easier for me to do that experiment, and I will do that experiment and let you know what's happening. But in the paper, we put it in bioarchive. We literally showed how the heart inflammation is reduced by disulfiram treatment. Please go and check that website. It is there. The data is there. It's unlikely it induces apoptosis in vivo, because we have never seen any apoptosis, which we can very clearly see after disulfiram exposure in these animals.